This is a production of Cornell University.
Okay, so hi everyone, my name is Elise Tomaszewski. I'm really excited to talk about my research where I've been using CRISPR-Cas9 to target fruit abscission-related genes in Physalis grisea, and I'll also go over an unexpected path that my research has taken over the last couple of years. So Physalis grisea, or ground cherry, is my model species. It's a species that I work on. It is a solanaceous fruit crop species, and it produces small, sweet yellow berries. However, it is an underutilized species. It's not grown on a large scale, largely because it is a semi-domesticated species, as it has a sprawling growth habit and high rates of fruit drop, hence the name ground cherry. The fruit are covered by an inflated calyx or a husk, and the husk of the color does not always correlate with the ripeness of the fruit. So on the left are fruit that have dropped, and I peeled back the calyces of all of these, and there is a clear gradient of ripeness in the fruit. So even though they have dropped, they are not always ripe. Not only is that an issue, but the fruit drop is an issue because you can then introduce soil-borne pests or pathogens to the fruit and making them unsellable, and farmers would lose in profit that way. Therefore, for my research, I am using CRISPR-Cas9 and Physalis grisea as my model species to explore the genetics behind fruit abscission, as well as trying to develop ground cherry lines with reduced fruit drop. There are 4 main steps of fruit abscission or fruit drop. However, the genetics behind each step is largely unknown. These 4 steps include the formation of an abscission zone, which are small cytoplasmically dense cells. This is where the detachment occurs, and it forms on the pedicel that connects the fruit to the plant. These abscission zone cells then become competent to external signals that induce cell separation, and then senescence and detachment occurs. For my research, though, I am interested in understanding the genetics behind the abscission zone formation and how that occurs. A likely group of genes for me to target were Mazbox transcription factors. These are largely known to upregulate or induce floral organ development, and they're also known to work in groups of 4 or a tetramer. In tomato, which I'm kind of illustrating on the left here, there are 3 known Mazbox transcription factors that induce abscission zone development. These include jointless, and what I'm showing is a wild-type tomato pedicel on the top and a jointless mutant pedicel on the bottom. There is no abscission zone. This is also the same for mutants of macrocalyx, or MC as my diagram shows, and jointless 2. However, interestingly, the jointless 2 ortholog does not exist in Physalis grisea, and I'll talk about that more at the end of my presentation. But for my work, I'm using CRISPR-Cas9 to target the ortholog of macrocalyx, which is MPF3, and the ortholog of jointless, which is also named jointless. And I hypothesized that knocking out these genes would also inhibit or not have an abscission zone form on the pedicel. And that is exactly what I found. So on the left is a wild-type ground cherry fruit, and zooming in, it's very clear where this abscission zone is present. This is where the senescence stops on the pedicel. This is where the fruit will detach. However, in my mutant in the middle here, this abscission zone does not exist. It's not present by the naked eye. In fact, these fruit will continue to stay on the plant and actually decay or continue to ripen until a physical force is applied. However, I do want to note that it's not impossible to pull these fruit off. It's not harming the plant like it would in a pepper or a goldenberry. Similarly to my jointless mutants, my MPF3 mutants also do not form an abscission zone, shown in the middle picture here as well. Another phenotype that I'm seeing on these MPF3 mutants only is that the calyx does not coalesce to a single point, as it does in the wild type. You can also see the berry in there. It's completely yellow, so it's already ripened, which is a great phenotype that I like to see, and it's helping with the uniformity of ripening. So just because I couldn't see the abscission zone by eye, doesn't mean that it didn't form, and I wanted to see on a microscopic level if the abscission zone was forming. So what I did was I chose fruit that the calyx or the fruit was starting to ripen. I embedded those in paraffin wax and sectioned those. And what I saw was an abscission zone in my wild type, so on the very left. How to orient yourself is that on the left would be the pedicel connecting to the plant, and towards the right would be connecting to the fruit. There is a clear zone of abscission zone cells. They're small and darkly stained. However, in my jointless mutant, shown in the middle here, this abscission zone is not present. I don't see these small banding cells. Similarly, in my MPF3 mutant, this also doesn't exist. This is exactly what I wanted to see. Zooming out, or on a larger scale, this is also important, both not only in a large-scale agricultural setting, but in the greenhouse. So I grew up wild-type plants, and my mutants, both jointless and MPF3, And these are at the same time, everything's the same. And you can see that there's large accumulation of fruit occurring on the grapes that hold up the plants. Not only does this cause more time for me as a researcher to have to go and clean my greenhouses, but you're increasing pressure of both diseases and pests in the greenhouse. Similarly, as I've already stated, on a large scale, this would be bad because you'd be losing profit due to fruit that are on the ground. So my mutants are holding up and they're dropping maybe 1 or 2 fruit, likely because you're bumping into the plant at that point. Up until this point, you guys have kindly assumed that I have created knockouts. However, the last couple of years, I've had a lot of issues detecting what edits I've made using CRISPR-Cas9. I couldn't produce an amplicon using PCR despite many different methods and attempts. And when looking further, I found that my region is rich in tandem AT repeats, which makes PCRing an awful nightmare. So I found a PCR protocol from Dotterwall and colleagues where they use a 2-step PCR method and they lower the extension temperature and increase the amount of magnesium chloride. Typically, an extension temperature of 72 degrees is used, and I couldn't get an amplicon shown by this gel here. So when I lower the extension temperature to 68 degrees, I was able to get an amplicon. So using that method and my optimized protocol, I finally got bands, which I was really excited for. So it's clear the band on the very right is my wild type, and all of my alleles are evidence of deletions in the gene itself. This is for jointless, by the way. However, this was also concerning to me because Physalis grisea is reported to be a diploid. So at maximum, I should be getting 2 edited alleles, and I'm seeing 3 bands in most of my samples here. This indicated to me that these weren't actually diploid, so I had to figure this out. And I did this with 2 ways. First, with a pollen germination screen that was adapted from the Littman lab, where they found that diploid plants had 1 pollen tube per pollen grain. However, non-diploids would have a mixture of 2 pollen tubes per pollen grain. What I'm showing on the left here is a picture of my wild type Physalis grisea pollen. And thankfully for me, these all have 1 pollen tube per pollen grain, meaning that my stock is in fact diploid. What I'm showing now are pictures of my mutants from jointless. So the top 2 here, they have these 2 pollen tubes per pollen grain, as well as this bottom left one here, indicating to me that they're not diploid. Also interestingly, my non-transformed control, or my wild type that went through regeneration, also had 2 pollen tubes per pollen grain. This is actually good for me because now I have a tetraploid wild type that I can use for comparison. I kind of already spoiled it a little bit, but I then needed to use flow cytometry to kind of figure out what was going on if I had an aneuploid, a hexaploid, or a tetraploid. So I was using leaf tissue with the help of Lena Wilson. And essentially what you can do with flow cytometry is estimate your ploidy level based on the nuclear fluorescence. So essentially a diploid would have half the amount of fluorescence as a tetraploid, and this would be shown by an increase on the x-axis. So on the left, you have a diploid peak at 50, and on the right, you have a tetraploid peak at 100. For my samples, this is exactly what I saw. So on the left is my wild type, and I had a fluorescence level of 10,000, but my non-transformed control, or the tetraploid wild type that I expected, was at 20,000, doubling the fluorescence peak, indicating to me that it was a tetraploid. This was also the case for my jointless plants, as well as my MPF3 plants. So before I go on to what's next, I also sympathize greatly with those who work with polyploid plants. This has been a lot of work, but I still have a lot more to go. I'm in the process of trying to identify other Matchbox transcription factors that interact with jointless and MPF3, seeing what might have taken the place of jointless too. I then will, once I get candidates, I plan to target them using CRISPR and also screen those for any abscission zone phenotypes. And with that, I'd like to thank all of my committee members, all of the members from the Van Eyck and Giovannoni labs, as well as the BTI greenhouse staff for keeping my plants healthy and happy. The flow cytometry team, especially Lena Wilson and Jackie Mahoney, as well as the Littman lab members, Sergey and Mopta from BTI for their help with the microscopy work, as well as Victoria and Tismark, who are my RNU students, Joseph Hill, who helped a lot with the PCR troubleshooting, and last but not least, Nathan Ream, who was a former member of the Van Eyck lab, who helped with the jointless CRISPR construct design, as well as generating the MPF3 mutants. And with that, I'm happy to take any questions. Yes, Ed.
Yeah, very nice. I was wondering, so is it easier to look at the pollen than it is to look at like root tips for ploidy, or?
I was also considering doing that. Because the protocol for Physalis and the pollen had already been figured out, it was a 3-hour protocol for me. So I could collect pollen, germinate it, and then I was looking in the microscope pretty like 3 to 4 hours later. So it worked well for me. I could use the root tip swash.
So about 3 hours.
Yeah, 3 to 4. Yeah.
Do you see any other types related to fertility in your paper?
Yeah, I don't want to be this person here, but we'll see. So let's see. While he's doing that, I promise they do. What really started this off for me was I was in the greenhouse and I noticed that my flowers were incredibly large. I thought I was kind of going crazy for a second there, but the flowers are very large, and if he's able to get this up, I'll show you. The other thing I noticed in the flowers themselves was deeper anthocyanin pigmentation. But you'll also have larger leaves. They're a lot greener, so I can pretty easily detect a tetraploid versus a diploid in my plants now. So if it ever comes back, you'll be able to see it.
There we go.
So not only are they larger, but they're also more pigmented, both in the petals and the filaments. This isn't necessarily a phenotype. I think this is a jointless phenotype, but yeah, I'm just showing the darker pigmentation here, and in the filaments you have anthocyanin pigmentation. Any other questions?
I think he's starting looking into levels. Sounds like
We don't really have any other Physalis grisea or ground cherry accessions that I could screen. I mean, we've screened the wild type enough that we have, and it's diploid. Our goldenberry, which is also a Physalis, is a tetraploid that we know and that people have reported on in the literature. But I have created other knockout lines that I'm not showing today. I think 3 of them. Almost all of them were tetraploids as well. I know other people in the lab have generated them as well, and it's about 80% you'll get that are tetraploid. So the high in Physalis at least. But I'll keep the ball moving here. Thank you.
Hello, everyone. Good afternoon. As you said, my name is Erin. I'm a third-year PhD student in Mike Gore's lab. And I'm excited to talk with you all today about integration of different proximal remote sensors for improved characterization of agronomically important traits such as yield. So proximal remote sensors have applications to both high throughput phenotyping used in breeding programs and by researchers for other projects, as well as precision agriculture. So traditional phenotyping methods are typically time and labor intensive and can be temporally constrained. High throughput phenotyping, on the other hand, may allow things like early season prediction of end of season traits, thereby decreasing the time of breeding cycle, can help with phenotyping more individuals for GWAS or training populations for genomic prediction. And being able to phenotype at greater temporal resolution may help understanding of genotype by environment interaction and parameterizing crop growth model. And then for growers, the ability to detect pest or disease and allocate resources to different parts of the field. However, application of proximal remote sensors to each of these things relies on both the relevance and the accuracy. Different sensors capture different spectra, and different platforms allow collection across varying scales. So there may be unique information contained within each of these that may increase both the relevance and accuracy of using these sensors. However, because of the complex and high-dimensional nature of these of the data streams collected from these sensors, integration is uncommon and typically lacks generalizability. So I'm going to be talking about 2 different platforms and sensors. First, we have multispectral images collected by unoccupied aerial vehicles. And then we also have LiDAR scans collected by unoccupied ground vehicles. So all of this data was collected on genomes to field maize hybrids planted at Musgrave Research Farm in Aurora. And these are all 2-row plots. Now, the multispectral images you can see here have 5 bands. We have blue, green, and red, which constitute typical image, and then you have red edge and near infrared as well. So the multispectral camera is mounted to the bottom of the drone here. And all of the raw images have about 80% overlap. So these are first stitched together using a software called Pix4D to create an orthomosaic. That orthomosaic is then uploaded to an online platform called ImageFree, developed in the Robbins lab, to take the orthomosaic and split it into these plot level images here. So the LiDAR scans are collected by the ground rover here. The LiDAR camera you can see on the back. The rover is driven in between the rows of the 2-row plots. And the LiDAR scans are converted into 3-dimensional point clouds. So you can see the 2 rows here and some additional points being picked up from adjacent plots, which are ultimately cropped out to give us this point cloud here. Now, each of these points has an X, Y, and Z coordinate denoting its position in space. So the LiDAR scans give us a 3D reconstruction of each plot. So in order to achieve integration, we're using machine learning models called autoencoders. So an autoencoder is a deep unsupervised machine learning model, which takes its input, for example, our multispectral image, passes it through a first neural network, the encoder, to produce what is called the latent space here. So this latent space is a reduced representation of the original image. This latent space is then passed through a second neural network, the decoder, which is analogous to the first, to reproduce that original image. And this model is optimized such that the difference between the original and the reconstruction is minimized. And in that way, the latent space or the latent phenotypes we hope the important features reducing the noise of these images. So we first trained this model on the multispectral images with an original dimensionality of about 40,000 values down to a single vector that is 512 values. So achieving integration on these images will involve training these models on multispectral images, and again on point clouds, extracting the latent space from each of these and concatenating them into a single object, which can then be used for prediction. So to evaluate training of the autoencoders on multispectral images and the point clouds, which I'll show in a moment, we looked at visualizations of the original images and the reconstruction. So you can see here that the latent space was able to effectively reconstruct our original image. So we similarly trained a model on the LiDAR scans. This involved a little bit or a couple of additional pre-processing steps. So we take the original point cloud, as you can see on the left, kind of stack the 2 rows together. So taking this x-axis and flipping it, and then compressing along the time dimension here to produce a singular single dimension density map. And again, you can see that the latent space contained both the sufficient and necessary information to reconstruct this high dimensional image. So the next thing that we wanted to do was do some preliminary investigations of what kind of information was contained within the latent space. So this is showing PCA plots of the 512 latent codes extracted from each of about 5,000 regions, colored first by NDVI and again by the date. So this is signaling that there is biologically relevant information contained within the latent space, and similarly do the same thing for our point clouds. We see a similar kind of clustering pattern. So our next steps are to take the latent spaces and use those to predict ground truth phenotypes. So we've done some preliminary tests of this with grain yield, which I've not shown here. Taking some simple linear models. We are seeing some biological signal, and in particular, we are looking at different ways to account for the temporal data that we have here. So with that, I'd like to thank my committee, my colleagues, Giovannoni, and Margaret Smith. I'd like to thank Liam, Nick Lepak, Amy Schultz, and others who have helped collect all of this data. And with that, I will take questions.
How do you measure that the reconstructed image is good enough to represent your original image?
That's a great question. So ultimately, we'd like to use the prediction accuracy from that second step. So once you've taken the latent space, using those to predict our ground truth phenotype. But these initial tests, looking at the loss of these different functions. I wouldn't. It's possible that more blurry or higher loss in the autoencoder would actually lead to a greater prediction accuracy of the phenotypes, less overfitting of the model. Ultimately, prediction accuracy.
Are you planning on looking at, I guess I'm curious when it comes to incorporating this into the prediction model and just using the drones versus the rovers, and one is more beneficial to the model than the other, or if it's really a combination of both that you think is going to give you the biggest opportunity?
Yeah, absolutely. So ultimately, I think that will depend on the trait itself. But for something like, more complex like yields or LAI or resources, the 3D architecture that that's something we are hypothesizing will lend additional information that will increase that accuracy. That is something that we're planning to test as well. So we just use the rovers, we just use the drone, we use them together. What does that look like? Done.
Can you go back to the slide with the PCA? I guess I was struck. There was the PC1 had explained 96% of the variance.
Yeah.
So, which suggests that all 512 are pretty highly correlated with each other. Am I understanding that right?
Yes, we were all surprised with the high.
So why not just represent this with, 2 values instead of 512? I mean, like that first PC is explaining almost everything that you have in your 512.
Yeah, so I think the advantage of not using something like PCA would be you don't have that linearity constraint on the looking at the relationship between the different values in the images. One potential reason that first value is so high is something like really highly correlated with the date or something. So you're really able to pick out like the brightness level. So even though that first PC is such a high percentage, it might be something that's not biologically relevant. But I think that's my opinion.
if you did the autoencoder process on just one date, so that date isn't a factor causing variation at the end of this, what would it look like? I mean, maybe this whole thing is swamped by date, in other words.
Yeah,
right?
that's helpful. That's something that I need to do.
So you can also get a 3D from the drone. What do you think was the figure for the like what do you expect to see from above once you talk about?
Yeah, so from below you're gonna get for your G reconstructions of like the ears themselves, for example, whereas from above, particularly later days you'll have that canopy closure that you're not going to be able to pick up on that. You know, things like stand count, yeah, other kind of that below canopy that when you have that canopy closure you're not reaching. And
Yeah, I was wondering if you tried going to autoencoder so it's trained, it's looking for heritability. So what you do is you train it not on the same row, you give it the paired replicated row in the other plot, and build the embedding space based on that.
yeah, that's a great point. Something that we have talked about doing but not doing yet is introducing, you know, like a secondary loss function for genotype or you know, spatial location in the field or something like that. So,
yeah, I actually actually make the result opposite compared to the replicated row and the other rep. Right? So,
Right.
yeah, I think that gets us kind of to extract out the genetic component behind the case.
Just so I can
Yeah.
question it. I'm like, in other words, you want the decoder to generate something that resembles the other replicate.
Right.
Yeah.
You can also do this by running the rotors going opposite direction down the rows and other side of the rows. So that's the other thing people are doing like food crops. They run down one side and they're trying to predict the other side.
oh, that's, yeah. Okay. Yeah. Thank you. Yeah, definitely gonna try. Yeah. Yeah. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.